It's now my pleasure to introduce our next speaker, Associate Professor Sue White. She is a clinical geneticist and the Deputy Clinical Director of the Victorian Clinical Genetic Services. Sue has a passionate interest in children with undiagnosed syndromes and works tirelessly with families to reach answers for their child's suspected genetic problems. Sue has led the implementation of the Exomes sequencing at the Victorian Clinical Genetic Services and the Royal Children's Hospital, the Undiagnosed Diseases Program in Victoria, and is a very worthy and valued ambassador of SWAN Australia. So please welcome Sue.
Thanks very much, and it's just great to be here at the SWAN conference. I'm really honoured to be able to talk to you today, and honoured to have my ongoing association with SWAN, and always grateful for the role that SWAN plays in supporting families with children with undiagnosed conditions or ultra rare conditions. So I got the easy job today. I get to just talk about what I do. David's done all the hard work of working you through all the science. So we're just going to kick back now and talk a bit about what clinical geneticists do. And I guess I've tried to weave into that some of the things that might be on your agenda that relate to what geneticists do. So what happens in a genetic clinic? Why do doctors do some of the weird things they do? I can't answer all the questions that might pertain to that, but maybe at least in the genetics clinic, what sort of tests we use. So I guess following on from what David's talked about, and where are we going? What do you do if you don't get an answer via a genomic test, that kind of thing. So we'll work our way through a bit of stuff and hopefully stimulate some conversation afterwards. So I thought I'd just start by talking a bit about what I do. So apologies for the Sue-centric focus of the next few slides. I work as a clinical geneticist at VCGS, Victorian Clinical Genetic Service, and I work as a clinician, so I don't have any funded research time. I work seeing patients. So there's a nasty photo of me in the top left corner in the clinic downstairs. Well, actually we're on the ground floor. In the clinic upstairs, it's downstairs from my office. So on the second floor, we see patients in the outpatient clinic at RCH. That forms a big part of my job. Yeah, we see patients several days a week in our outpatients, and we also see inpatients. So babies and children and teenagers who are in the hospital because they're unwell for some particular reason, and their medical team think they might have an underlying genetic condition. Those patients get referred to us as inpatients. So this pretty cute baby up the top here who is an inpatient might be the type of patient that we might see. Interestingly, our job as geneticists, I guess when I started a long, long time ago training in clinical genetics, pretty much that sort of clinical role, that was pretty much everything that we did. We saw patients, we went on ward rounds and saw inpatients. We attended clinical meetings with our colleagues. That was sort of pretty much it. But with genomics, our role has really changed enormously. And so now a fair amount of my week is also taken up with meetings, very close collaboration with our laboratory colleagues, looking at genomic data, trying to make sense of it. So David already mentioned in the question session about how important it is taking the clinical information and trying to marry it with the genomic data to get meaning from it. And so, in fact, we do a lot of, excuse me, we do a lot of that kind of work, both for our own patients that we see, but also any patients that get referred to our laboratory, where we're looking at the genomic data and trying to make sense of what it actually means for the patient. Most consultant clinical geneticists will also play a role supervising both trainee geneticists and also genetic counsellors who are really valuable members of our team. And these two particular smiling genetic counsellors are currently playing a new role in the hospital as a laboratory liaison genetic counsellor. So what they're doing is looking at all the requests that come in through the hospital laboratory to try and make sure that the right patient is getting the right test. And that's a step removed from you and your child in the clinic, but it's a really important background role, because there's no point doing the wrong test on a patient, on an undiagnosed patient. It's really important to ensure that the patient is getting the tests that they need in order to maximise our chance of making a diagnosis, both from the best care for that child, but also from the point of view of spending our precious health dollars wisely. And part of that has meant that we also spending a lot of time trying to upskill our non-genetics colleagues. So within the hospital, we have specialist paediatricians, surgeons, emergency medicine physicians, and for most of them, genetics is a pretty peripheral part of their job, right? But genomics is having an impact on specialists of all kinds of persuasions. And so our job as geneticists has evolved to become an education and advice role with our colleagues who are trying to work out how do they integrate genomics into how they work as a neurologist or as a cardiologist. And some of you will be seeing those types of subspecialists, and that's really important work that they're doing. So we do venture outside the hospital occasionally, and most clinical geneticists, as well as their metropolitan role, will do some regional clinics. So my regional clinic is in Shepparton. So 3 times a year I jump on the train with a trainee geneticist or a fellow and a genetic counsellor, and we head to Shepparton to do a 2-day clinic there. We do clinics in about, I can't remember the number, but maybe 8 or so regional centres around Victoria. And increasingly too, we're playing a role working with other specialist groups. So I mentioned that lots of our non-genetics colleagues are trying to learn how to use genomics in their clinic. So some of the different units have taken on different models, but with the kidney team, we've ended up forming a multidisciplinary clinic where there is a geneticist, a genetic counsellor, and a kidney specialist, all seeing patients, kind of supporting each other, getting advice from each other to help children who particularly have some sort of kidney problem that looks like it might be genetic. And that's been a really interesting model, and I've learned a lot about kidneys in the process. I supervise our trainee clinical geneticists, and so some of you might see them as your doctor when you come to the genetics clinic, and here they are looking very happy because they're at a genetics conference and having a bit of downtime. I just had to include that photo because it's kind of funny but not really relevant. But I think I wasn't actually at this conference, but I think that it was in an art gallery or there was some session, and this was some kind of sculpture of a heart or some anatomy. Anyway, Lil, who's one of our fellows, looks pretty surprised to be popping out of the pulmonary artery or whatever she's doing there. And you might recognise this interloper is the head of our genomics lab. So he's a good person to know if you're looking for a genomic test. So these are the next generation of clinical geneticists, and we really need to support them and teach them about genomics and clinical genetics in order that they can meet the needs of your families and the families to come. So genomics does take quite a team of people to deliver. It's quite a cumbersome and ambitious, and what I would say is kind of disruptive technology. By that I mean it doesn't just slip really sort of seamlessly into the way that we all work. There's all these new roles we have to sort of create and carve out. There's a lot of, as David mentioned, there's masses of data from which we're trying to get, crystallise some amount of meaning or message to give back to you as families, or to give back to the doctors who have referred you. And so it's really taking us time to integrate it into the way we work. So we've had to change very much the way we work, and to make sure that we can deliver it in a sustainable way. And clearly we've still got lots of work to do in that regard. So it's generated as well a lot of acronyms, because we have all these new meetings that we have to go to. And of course, I don't know what industries you all work in, but we seem to have a lot of acronyms in our industry. So we go to VPCs, which are variant prioritization clinics, which is where we have clinical geneticists and fellows and scientists from the laboratory looking at the genomic data from an exome or other genomic test, trying to work out which variants need to be carefully curated by the laboratory, which variants might be important for this patient, of the thousands of variants that the test has generated. We have MDTs, which I know happen in lots of different areas, but in our area, multidisciplinary team meeting is where the lab's gone away and curated those variants, and they come back and we debate, how are we going to report this? Is this a pathogenic variant like David was talking about? Is it a benign variant, and in fact we don't even put it on the report? Or is it one of those pesky VUSs, the variants of uncertain significance that we all dislike intensely but are a reality of genomic testing? We're stuck with them for the time being, and where the clinician has to try and work out if they're relevant and how to take forward further investigation. And then there are a whole bunch of other sort of political meetings about how do we make genomics work across the state, and research meetings that I'll tell you a little bit more about if I better get my skates on though, because I'm conscious I'm going to run out of time, aren't I, Linda? Yes, she's saying, stop talking so much. So these photos are just photos of the crowd of people who are working on genomics at VCGS, and you know, there are scientists, bioinformaticians, genetic counsellors, clinical geneticists, fellows. It really takes quite a bunch of people. All right, that's enough about me. Now I'm going to talk about some of the questions you might have of a clinical geneticist, and how you interact with the genetics clinic to get the best for your child. And perhaps we can talk some more about this if I don't fully answer your questions. But Heather did prompt me with a few questions that, you know, she gets asked a lot that perhaps I could tackle. How do you ask for a genetic test? And can you self-refer to genetics? And I'm a bit hesitant here because it sounds a bit like I'm telling you what to do. So please take my comments as just a few ideas that come from one side of the fence, and you will have some other ideas about how you navigate these tricky areas of being an advocate for your child, a parent for your child, but also I guess you're trying to work within the relationship that you have with your doctor. I think it's really important to engage in a dialogue with your specialist, and I've used paediatrician here, but it might be a variety of different specialists, about your desire to understand what is going on. Most genetic services do prefer to have a referral, ideally from a paediatrician. We do accept referrals from GPs, but I guess we're often after quite specific information about the pattern of problems in the patient, and GPs have not always got that depth of information that we're after. Some services who bill under Medicare need a referral to see your child for billing purposes. So the easiest way is to get a referral, if you can, from your paediatrician or your specialist. I guess it's worth thinking all the way through about you know, the upsides and downsides, or things to be aware of around genetic tests and what genetics can offer you. There's no doubt there are these amazing, powerful new genomic technologies which are delivering diagnoses, as David said, in quite staggering rates. And these tests are becoming more integrated into standard clinical care. And in the next 5 years are going to move beyond clinical geneticists to lots of non-geneticists ordering the test. So I mean paediatricians ordering genomic tests. So I think they're positive developments from the point of view of access. There's no doubt about it. You do have to remember some unfortunate realities, I guess. One is that not all unexplained health and developmental issues have a genetic basis, number one. And number two, not all genetic type problems are suitable for genomic tests. So some problems might have a genetic contribution to the cause, but they may not be a pure genetic condition that a genomic test can diagnose. That's a difficult concept, but I guess what it means is that not every undiagnosed child is going to be suitable for a genomic test, even if we had limitless funding. And it's true that delivery of genomic tests is still more fragmented than we would like. We have made massive gains, but there are absolutely gaps in access and a lot that we continue to work on to try and ensure equity of access to genomic tests. And one contributing factor, but not the only one, is that practitioner awareness of what genomics can deliver varies. And so if your child doesn't or has a clinician who is not very aware of the promise and power of genomics, that might make it more difficult for you to access a genomic test. Perhaps we can talk about that conundrum a bit more later. So what do we do? How does a geneticist make a diagnosis? It really is, I think a jigsaw is a good analogy. It really is a piecing together of the pattern of problems in front of us that we're trying to make sense of from a genetic point of view. So I've written perhaps just a list of problems here that might occur in a child who might come along from a paediatrician with a cleft palate, so a hole in their palate, a small chin, a hole in their heart, short stature, and maybe facial features that are a little different to their family. So that cluster of problems, a combination of malformations in the heart and in the palate and some features that are unique and distinctive, makes us wonder about a genetic problem. So that's a first part of our jigsaw that we're trying to piece together for this child. So we often in those situations will undertake various tests and also use photographs. And families sometimes ask me, you know, what is it about this photograph business? What are you doing? Because I guess it's not a common part of clinical medicine to take photographs of a patient. We really use photographs to allow or to enable that child to essentially be assessed by a group of clinical geneticists. So every week we have a meeting where we sit down and we review photographs of patients who are undiagnosed. And together we try and do our best to see if we can make a diagnosis or work out what are the best next steps for that particular child or adult. We also look at photos of kids and adults who have recently got diagnoses to try and learn about those and implement them more into our practice. So the photographs are typically used in a medical multidisciplinary meeting where we're trying to advance the care of that child. We often will go imaging other organs because if a child's got a cleft palate and they've got a hole in their heart, they may have abnormalities in other organs that we can't see. So their kidneys or their brain, sometimes we'll ask for an eye examination. So we're trying to piece together, what does this jigsaw look like? Is it a recognisable picture that we look at and go, oh, I know what this is? Or is it something that we think is genetic? And a lot of you might have had this conversation with your doctor. I think the problem's genetic, but I don't know which gene it is. And that's because, as David mentioned earlier, there's more than 1,000 different genes, for example, involved in causing intellectual disability. So it's unusual for us to be able to say, well, I know for a fact that it's this one particular gene out of that particular 1,000. We look in databases. There are medical databases like POSSUM, which is a MCRI-hosted database where we put in search terms like this list and try and see if we can find a diagnosis. We use open access databases like OMIM, and we use Dr. Google just like you do when we're desperate. And genetic tests, I guess, is something that we are talking about a lot this morning, and I'm going to cover off a bit further, so I'll jump ahead. So what might you expect when you walk in the room? We typically, for a new genetics appointment, we might take 45 minutes or an hour for the appointment. I think sometimes the appointment with a geneticist can feel like it's quite an in-depth appointment, and I chose that picture quite deliberately because I think there's a lot of scrutiny of the geneticist on the child and the family in the appointment, and I think it's good to understand why we're doing that and what purpose it serves. So we ask about your family history and we draw a family tree. We ask about the pregnancy, and we're trying to look to see, are there other clues to add to our jigsaw of the picture of your child? When we're examining the child, we are looking for unique features, and every child, of course, their 23,000 genes have come from their parents, and there are all kinds of features that we all inherit as just familial, facial and other features. And so we're always trying to look at this child in the context of their family. What features are just running in their family? What features might be part of their underlying genetic condition if they have one? And so we do compare features with other family members in the room, either in the room or by photograph if not. And then I guess we try and leave enough time to have a discussion about how likely we think a genetic diagnosis might be and what tests might be possible, how are they done, the annoying and challenging issue of what is the cost, who's going to pay, how long do they take, and how likely are you to get an answer from this test. And hopefully we come away with a plan that's clear to you and to us. Typically in genetics, we write a letter back to the family. That's been a practice for a long time. And I guess it reflects a strong desire by us as clinicians where our job is communication. And I think the letter's a very important part of that. So one really important thing to understand is the difference between a clinical test and a research test. So David talked a lot about different tests earlier. We've talked a bit about microarray. Often Fragile X will be ordered in a child with developmental problems. We might do gene panels. We might do a singleton exome, which is in the child alone, or we might do a trio exome, which is the child and the parents. And we also have access to a lot of tests that are in the research realm. I guess the key thing for you to understand as families is that clinical tests are done in laboratories that usually have accreditation from a laboratory organisation, which is usually a national accreditation, which enables them to issue a clinical report that goes into your child's medical record. Research laboratories are working in much different sort of parameters. They are often, their funding is often a lot more precarious and goes from grant to grant, that they often won't be able to give you a necessary turnaround time for a test, and you may or may not get a research report from any kind of research testing. Ideally, all diagnostic findings from a research setting would be validated in a clinical lab, but funding can often get in the way of that. So I guess just something to be aware is we are working in a really rapidly changing field, and so access to tests, the cost, the yield can fluctuate from lab to lab, from service to service, and even within a service over time. So the advice that we're talking about right now will be different in 6 months. Tests on offer, the cost and the funding mechanisms change. I might skip over exomes versus panels just in the interest of time because David covered it off a bit earlier. We can come back to it if you're interested. What's the fuss about exomes? Why are we so energised by them, and why are they so powerful? As David mentioned earlier, they yield a diagnosis in children with suspected syndromes, carefully selected, I should say, in more than half of the patients. And this is an important part, 5 times the rate of the standard care those patients got. So what that tells you is that the background diagnostic rate in that patient group, which is really the SWAN families largely, was pretty poor. You guys knew that already, right? But I guess that study that we undertook demonstrated that. And what we learned was that when we made a diagnosis, care changed in 1 in 4 of those children. We know we can't cure genetic conditions, but we do a better job of looking after children with suspected genetic conditions when we find out what that condition is, by stopping or starting surveillance, stopping or starting medication, and understanding the natural history of that condition. We found that it was a very cost-effective test. When we did the study, it was cost a quarter of the price of standard care per diagnosis. The cost of exomes has increased, but it's still, an exome costs half the price of standard care per diagnosis on 2019 prices. But you reap the benefit when you do it early in the diagnostic pathway. And so this slide just shows the difference between standard care, which gave us in the infants in this particular study of children with undiagnosed syndromes. Standard care gave us a diagnosis in 11 out of 80, whereas the exome in the same patient group gave us a diagnosis in more than half. So why doesn't it always work? What if you've had an exome and you didn't get anything useful out of it? There are a whole heap of reasons, unfortunately, why your child's exome might be negative. Some of them relate to the limitations of the test. So the test cannot detect all DNA changes within the coding regions of a child's genetic material. So it's not a perfect test by any means. It's a great test, but it's imperfect. So there are gaps in the sequencing coverage. There are also genetic conditions or situations where the test cannot easily or readily detect the type of DNA change. So one of those is called mosaicism, where a child has a DNA change in some cells but not in others, and that can make the test, it can make it really difficult to find that DNA change depending on what source of DNA you're using for the test and some of the technical specifications of the test. David talked earlier about how we have 23,000 genes, but we probably have about 5,000 that we know, in which we know mutations lead to certain types of health problems. So we call that the Mendeliome. Another bit of jargon you can see if you can slip into your dinner time conversation today. So the Mendeliome is that collection of 5,000 genes that we know the gene-disease association. So there's a great big bunch of genes, thousands of genes. We don't know anything about what changes in those genes are. So your child's exome might be negative because the damaging DNA change might be out here, and we won't know how to analyse that. Clinical exome sequencing focuses on genes we know something about. And there are lots of things about how DNA can be different that are not well captured by genomic sequencing. So David talked about single nucleotide variation where one DNA base is changed, but there are lots of other spelling mistakes, things called triplet repeat disorders, other ways your DNA can be mucked up that genomic sequencing really struggles. How are we going for time? Oh, I'm seeing an orange thing. That's okay. All right. All right, I'll do an orange thing version of the last few slides. What's on offer? What's possible if your child's exome is negative and we still suspect there's a very likely genetic diagnosis? Just to mention a couple of programs that are being run here on this campus. One is the Undiagnosed Diseases program, which really tries to take patients who have had that clinical exome, didn't get a diagnosis, but their doctors still firmly believe there's a genetic diagnosis to uncover, and to try and do some research-based genomic and other testing to try and get to the bottom of it. And so we have some early sort of results from this program. I think it's been running for maybe 2 years or so now, and the first nearly 100 families through. And what we found was diagnoses, I think, in nearly half of these patients, some in known genes already. So some within that Mendeliome, if we go back to that other picture, and some in new genes. So both those groups, I guess, showing us that genomic testing, clinical genomic testing has its limitations, but also there are still plenty of new genes for us to discover and a lot of work for us to do. We use different types of technology with that: trio exome sequencing, whole genome sequencing, and RNA sequencing, which is looking at the molecule that's sort of like the intermediary between the DNA chemical code in the nucleus and making a protein that's going to be the end result of that DNA. So sometimes we can unlock a child's diagnosis by looking at the RNA, that intermediary step, and try and see if there is information in there that gives us a clue as to what gene is involved. And quickly to finish off, rare disease at MCRI, there's a lot of work going on this campus to look at how do we do research better in the rare disease space. So this crazy solar system picture is really meant to show that there's a lot of different groups doing rare disease research on this campus, and you'll see here a lot of outcomes from that, but actually we want to do better at making that a system to deliver rare disease research, really to maximise opportunities for families to be involved and maximise what we're after, which is to enable all people with a rare disease to get a precise diagnosis, have appropriate care, and if it's available, have access to any therapy. So we've got a lot of work to do in that regard. That's work that's ongoing and being led by Kaz Simons, Tiong Tan, David Thorburn and myself. A couple of last slides, because I know I'm probably stretching my friendship here by still talking. What about support after a genomic test? I know this is a topic that's passionate and close to Heather and the SWAN team's heart, because they provide a lot of that support. And we're very conscious that delivering a diagnosis, as I think someone mentioned earlier, you can get a diagnosis but sort of still feel in limbo, especially if your diagnosis is ultra rare or you're the only family that you're aware of. There's 2 projects currently underway where we're exploring support needs of families with undiagnosed conditions, but also ones who get a genetic diagnosis, because we know there's a lot more we need to do in that space. Alrighty, I promise I'm pretty much there. Genomic tests are a powerful breakthrough test. I hope we've convinced you of that. They don't solve every problem. Our field is on the move. Come with us, but understand that things are moving all the time. And I think perhaps a good strategy is to work with your health professionals to understand what's available for your child. And I hope we continue to give more access to diagnostic tests for SWAN families. Thanks very much.
Thanks, Sue. So we'll just take one question now. And Sue, if you're willing to stay around during break, if you'd like to come and mingle with Sue, where the refreshments are being held, and ask your other questions then. So just one question now. Anyone going to take it? Yep.
Desperate for coffee.
Kate?
Thanks so much for that presentation. A quick question about singleton, exome and trio testing. When would you go for a trio rather than just a single child test?
Thanks for the question, and I think it's an important thing to understand. I think the message, so singleton is where you just test the child or the person with the condition, and a trio is where you add the DNA, you sequence the parents as well, and you're comparing the child's DNA to the parents. And I think there's a lot of sort of perhaps a bit the same as exome versus genome, singleton versus trio. I think people can sometimes feel like, oh, I want the best test, so I want a trio rather than a singleton. I want a genome rather than exome because more must be better, right? I think what we know from studies we've done is, in fact, both tests work well as diagnostic tests. Actually, a trio has a few advantages for us at a system level, from an efficiency level of shortening the time that it takes us to analyse a particular test, and also giving conclusive answers to the families, because it resolves some VUSs by looking at the parents' DNA. We can immediately chuck them out, or we can immediately know that they might be important. But rest assured, if you've had access to a singleton exome, you've had access to an extremely good current diagnostic test. And in our studies, there's no significant difference in the diagnostic yield. Sometimes we find that in different laboratories that can vary, because it does depend on the way the two different tests are handled. But they're both good diagnostic tests in the long term. To manage the number of people who need genomic tests, we need to do more trios. And that's just because we don't have enough. The singleton analysis is still very laborious, and too much hand waving. And so we need, from a sort of efficiency and system point of view, to move towards more trios to enable more tests to get through in a timely manner.
Thanks, Sue, very much. And Sue will be hanging around for further questions.